everyone. In this video, I will show you how to conduct the reverse osmosis experiment. The video lacks some details, so please make sure to read the lab manual carefully as well. I'm standing in front of a vertical centrifugal pump, which is one of the main equipment. I will first explain the equipment and tools, followed by safety precautions, and finally, experimental procedures. This is a diagram showing the overview of the reverse osmosis experimental apparatus. Start by tracing the lines and valves in the apparatus. Try to understand the purposes of the valves. This is a suction valve located at the pump inlet. It's a discharge valve located at the pump outlet. The vertical centrifugal pump has multiple impellers in the column to deliver high pressure. Pump's prime inlet is located at the upper right side, and you need a wrench to open the screw. The pump's prime outlet is located at the upper left side, and you need another smaller wrench to open the screw. At the top of the pump is a motor in the black housing. Between the motor and the stainless steel column containing fluid, there is a joint shaft which spins fast behind the metal shield. This is an electrical switch, a flip switch for the centrifugal pump. The centrifugal pump uses a special electrical outlet. We use a 10-liter carboy during standby, startup, and cleaning of the pump. We use this gray bucket as a reservoir containing fresh water or salt water to be fed to the pump. The stainless steel column lying on the bench is a membrane housing. The RO membrane is inside the housing and cannot be seen. On the right end of the membrane housing, there is a filter inlet which is out of center, and a back flush inlet which is at the center of the cap. On the left end of the membrane housing, there is a reject outlet which is out of center, and a filtrate outlet at the center of the cap. Once water is discharged from the pump, it normally goes to the filter inlet if the back flush inlet and the bypass valves are closed. If the bypass valves are open, then water can be diverted to the bypass lines instead of going to the filter. On the back end of the filter, water can either flow to the filtrate line or reject line, more or less. There are two pressure gauges at the front end of the filter. One is for filter inlet line and the other for the backwash inlet line. There are two pressure gauges at the back end of the filter. One is for the reject line and the other for the filtrate line. There are two TDS meters at the back end of the filter. One is for the filtrate line and the other for the reject line. The TDS meters can qualitatively monitor salt concentrations. A gear pump and a stir plate are stored under the bench. You can use a balance located on a separate bench. The RTD meter and the offline TDS meter are stored next to the filter housing. You should find a glass funnel, beakers, magnetic stir bar, a plastic jar, and a 20-liter carboy with DI water nearby. Sodium chloride can be found in an overhead cabinet above the filter housing. There is a sink on the left side of the filter housing where you can dump water and get fresh tap water. Even though salt water is relatively harmless, you are still required to wear safety goggles and disposable nitrile gloves. Make sure to read safety reminders on the wall next to the centrifugal pump. Do not touch the pump motor or the metal shield covering the rotating shaft. Do not pull or push the pump, even though pump is fixed to the wall with a bracket. Do not move the bench where the centrifugal pump is located. Do not handle the electrical plug with a wet glove. You might get electrocuted. Make sure you wipe your glove. This experiment uses relatively high pressure that you can check with the pressure gauge. Do not wiggle the connectors unnecessarily. Avoid inlet pressure over 100 psi. Open bypass valves to relieve excessive pressure. Make sure to tighten the prime inlet and the outlet screws. Otherwise, water will jet out when you turn on the pump. If you find a consistent water leak, even if it's small, notify the instructor and TA. Make TDS meter calibration solution. Prepare a high concentration stock solution of sodium chloride in a large plastic jar or a large beaker first. The balance to weigh sodium chloride and a magnetic stir plate to dissolve it. Once you make a high concentration stock solution, you can make low concentration solutions in small beakers via serial dilution. In serial dilution, make sure you mix the water and the parent solution completely in a beaker before you move on to the next dilution. Use a glass bar to mix, and after mixing, you can aliquot a small volume to a small beaker for TDS measurement. You should use a TDS meter labeled offline. Hold the probe and stir the calibration solution for a while. Then stop moving the probe and wait until the measured ppm value stabilizes. Empty the carboy into the sink. Open the prime inlet with a wrench. Open the prime outlet with another wrench. All 5 tubes are inserted in the carboy and open the red suction valve. In the pump discharge valve and bypass valves, this will initiate the pump draining. You should see the water flowing out of the pump through the tubes. In the sink, fill the bucket with tap water to about 3/4 of the height. A bucket on the bench next to the centrifugal pump. Then place the gear pump between the bucket and the centrifugal pump. Plug in the gear pump. Insert the suction tube of the gear pump and one end of the small plastic tube into the water in the bucket. There is a black plastic connector at the end of the gear pump's discharge tube. Hold it with the end facing away from you. Twist the tube counterclockwise a few rotations, and then insert the connector into the prime inlet. When you screw in the black connector in a clockwise rotation, it may not go in smoothly. You may feel resistance, and the connector is angled relative to the inlet port. If that's the case, you must start over. When the connector is inserted correctly, it goes in smoothly without resistance, all the way to the end. While one end of the small plastic tube is in the bucket of water, you need to hold the other end and press it into the prime outlet. Make sure to hold one end of the small plastic tube and the suction tube of the gear pump so that they are submerged in the water. Flip the switch of the gear pump, dial up the black knob past halfway, and wait until water goes up to the prime inlet. Then close the suction valve and discharge valve of the centrifugal pump. As you fill up the centrifugal pump with water, initially you will see bubbles coming out of the end of the small tube in the bucket. After a while, when the centrifugal pump becomes full of water, you will see water coming out of the prime outlet through the small tube. Wait until water flows steadily in the small tube and no bubbles come out. Then turn off the gear pump. Keep pressing the small tube into the prime outlet. The residual pressure will push some water out. Hold the tube there until nothing comes out and it becomes quiet. After you remove the small tube, wipe the prime inlet and outlet for any water spills. Move the black plastic connector from the prime inlet and close the prime inlet with a plug and a wrench very tightly. Close the prime outlet with a plug and a wrench tightly. Hold the discharge and suction tubes of the gear pump over the bucket, pointing to the water surface. Turn on the gear pump at a high speed for a while to run it dry to remove water, and then turn it off. In the sink, fill the bucket with tap water to about 3/4 of the height again. Move the bucket next to the carboy on the floor. Move the two long tubes for reject and filtrate lines from the carboy to the sink. Move the pump suction tube from the carboy to the bucket. Then move the bucket onto the white tray, which is a secondary containment. Empty the carboy into the sink. Move back the carboy and insert the bypass tubes. Hold the open end of the centrifugal pump suction tube at the height of your face. Fill the tube with water using a funnel. Open the pump suction valve and air bubbles may come out of the tube. Then add more water to fill the tube again. Close the suction valve. Close the opening of the suction tube with your thumb. Bring it to the bucket, submerge it underwater, and then release your finger to keep air out of the tube. Keep the opening of the suction tube submerged. Attach the yellow filter to it underwater and make sure the yellow filter lies flat on the bottom of the bucket. Make sure the filter inlet valve and two bypass valves are open, while the back flush valve is closed. The two valves after the filter housing should be fully open as well. Plug in the electrical plug of the centrifugal pump in the special wall outlet. Turn on the pump by flipping the switch, and you will hear a loud noise of the pump motor. Open the pump suction valve and then pump discharge valve. Make sure water is flowing in the bypass lines. After a while, close the bypass valves one at a time and make sure the filter inlet pressure at the gauge 1 increases. When the bypass valves are closed, more water will flow out of the filtrate line tube and reject line tube in the sink. Keep the water level in the bucket above halfway by continuously filling, transporting, and pouring water in the plastic jar. Use two plastic jars to fill and pour alternately. Open the backwash inlet valve to flush both sides of the membrane filter to remove air bubbles and dirty things, if any. Close the backwash valve after a while. This step usually increases the filter inlet pressure. Make sure to achieve the filter inlet pressure of 50 psi or above by closing the bypass valves only. Turn on the two inline TDS meters. Make sure the filtrate TDS meter 1 shows below 20 ppm and the reject TDS meter 2 shows below 100 ppm. If not, keep adding tap water to the bucket and keep flushing the pump and filter. When you achieve the pressure and TDS reading goals, open the bypass valves to flush the bypass valves with fresh water. Turn off the centrifugal pump by flipping the switch, and immediately after that, close the pump suction valve and then pump discharge valve. Move the filtrate and reject line tubes from the sink to the carboy. Removing the yellow filter, move the pump suction tube from the bucket to the carboy. Empty the bucket into the sink. Pour 2 liters of tap water in a plastic jar and pour it into the bucket. Repeat this until you have 10 liters of water in the bucket. Move the bucket onto the white tray on the floor under the bench and move all 5 tubes from the carboy to the bucket. Empty the carboy into the sink. Scoop water from the bucket using a plastic jar. Hold the open end of the centrifugal pump suction tube at the height of your face. Fill the tube with water using a funnel. Open the pump suction valve and air bubbles may come out of the tube. Then add more water to fill the tube again. Close the suction valve. Close the opening of the suction tube with your thumb. Bring it to the bucket. Submerge it underwater and then release your thumb to keep air out of the tube. Attach the yellow filter to the suction tube underwater. Then pour remaining water in the jar back to the bucket. To prepare 2 liters of a concentrated salt solution, weigh 16 grams of sodium chloride using a balance. Pour 2 liters of tap water in a plastic jar. Dissolve the sodium chloride into a jar of water on a magnetic stir plate. After making the salt solution, turn off, plug off, and return the magnetic stir plate to under the bench. Turn on the centrifugal pump by flipping the switch. Open the suction valve and then discharge valve. Then quickly open the bypass valves. Add the concentrated salt solution to the bucket. After 2 minutes of recirculation through the bypass loops, close the bypass valves to adjust the filter inlet pressure to 50 psi. Keep the filter inlet pressure at 50 psi. The backflush pressure should be almost zero. On the back end of the filter housing, the reject line pressure should be almost 50 psi, and the filtrate pressure should be almost zero. The reject line TDS meter 2 should be somewhere between 800 to 1100 ppm. The filtrate line TDS meter 1 should be below 100 ppm, preferably below 50 ppm. Select a filtrate sample using a small beaker. The filtrate line tube is marked with one line of black tape. Make sure to shake off the tube over the bucket and wait a little bit before sampling to avoid contamination from the high concentration feed solution. Collect a reject sample using a small beaker. The reject line tube is marked with two lines of black tape. Collect a feed sample directly from the bucket using a small beaker. Make sure to be away from the ends of the reject or filtrate tubes. Use an offline TDS meter to measure the ppm values of the three samples. Shake the probe a little bit in solution and wait until the reading stabilizes. These values give accurate salt concentration compared with the inline TDS meter. Using a stopwatch and a graduated cylinder, measure the flow rate of the filtrate. It is ideal to do this with two people, one person holding the graduated cylinder. Measure the reject flow rate in a similar manner. Use a bigger graduated cylinder because the reject flow rate is higher. Using an altimeter, measure the temperature of the feed water in the bucket. Make sure to shake the probe in water for a while, and then stop shaking. Wait until the temperature reading stabilizes. Once you are done with sample collection and measurements for 50 psi, close the bypass valve more to adjust the inlet pressure to 70 psi. Wait for a few minutes until new steady state is reached. Then repeat the sample collection and measurements. Likewise, repeat the sample collection and measurements at 90 psi. Throughout the data collection, monitor the inlet pressure as it tends to creep up. Do not exceed 100 psi. Turn off the centrifugal pump by flipping the switch. Close the pump suction valve and then discharge valve. Finally, plug off the electrical cord from the wall outlet. On day 1, TA will clean the RO system for you after the experiment. On day 2, you basically repeat the same experimental procedure with a doubled salt concentration. If the calibration curve for the TDS meter is good for day 1 experiment, you do not have to repeat on day 2. At the end of the day 2 experiment, you will be asked to clean the RO system. The cleaning procedure is basically the repetition of the priming and flushing procedures.